Good afternoon, and thank you for joining. I'm Christine Happel, a Program Manager with NCATS, and I welcome you to today's presentation as part of the Extracellular RNA Communication Consortium webinar series. The ERCC is an NIH Common Fund program that works to advance the science and research of extracellular RNA. Stage 1 of the consortium started in 2013, with the second stage starting during September of this year. The consortium hosts monthly presentations on a variety of research topics, and I'm very pleased to introduce today's speaker. We have today Dr. Anastasia Khvorova, a professor, RNA Therapeutics Institute and Program in Molecular Medicine at the University of Massachusetts Medical Center. The title of Anastasia's presentation is Engineered Exosomes for Neuronal Delivery. And with that, Anastasia, I'd like you go ahead and take over.
Okay. Thank you so much for inviting me to give this webinar. And my lab, the work I'm going to present today is a very tight collaboration between different entities, which include working very closely with Nila Running Lab. It's another lab in the RNA Therapeutics Institute. It's a Marian De Figlia lab in MGH, and it's also a collaboration with Berk, which is a company doing a lot of proteomics and lipidomics studies. And what I would like to emphasize that the project internally was mostly driven by a very talented MD PhD student, which is now doing her residency in Germany and actually studying her lab later this year, Rekha Farasti. So we decided the goal of the project was trying to understand the molecular mechanistic footprinting or underpinning behind ability of exosomes to traffic information between the cells, which I think right now is a clearly established fact. And what we were interested in particular is to see whether we can learn how to use those vesicles for the delivery of therapeutic oligonucleotides, in particular sRNAs. And the four key questions which we wanted to answer is how to load the sRNA cargo, what is the most efficient mechanism of how you can get it associated with the exosomes, how to produce exosome on scale and quality. We also wanted to see whether changing the cell line identity or cellular conditions or media condition in which cells are grown will affect the physical composition and property, and also try to venture in a directions of linking the composition of extracellular vesicles to their ability to traffic oligonucleotide inside the neurons. Okay, so how to load the sRNAs and other nucleic acids in the cells? There are fundamentally two different approaches, and the approach on the left is manipulation of the producer cell, where you can, and this has been reported by several labs, including Cassandra Labs in Harvard, is introducing zip codes and binding motif which enrich association of RNA with the exosomes. It can be tethered to proteins, and alternatively, you can just transfect RNA inside the cells with this idea that some of it will eventually make it to exosomes. The other directions which was explored is manipulation of isolated vesicle itself. And the most common path which is utilized a lot, it's electroporation of exosomes as well. but there are publications both stating that this methodology can be effective, or alternatively that this methodology actually can destroy the exosome property. It's transfection of RNA in the exosomes, and while this is functional, the major issue with this approach that you're introducing lipids in vesicles itself, which can dramatically alter its properties. And then association with the membrane, which actually our lab was first to pioneer. So what we have been working in the lab a lot is fully chemically stabilized hydrophobic sRNAs, and in this case, the RNA backbone is substituted by a wide range of modifications, fluor, metal, chemically stable mimics of the phosphate, phosphorothioates, which is substitution of non-bridging oxygen in a phosphate backbone. And combination of those chemical modifications allow to develop molecules which are very metabolically stable, but simultaneously efficiently enter into RISC complex and can silence genes in neurons and other cells. And their uptake in the cells is driven by two factors, is conjugating this hydrophobic entity, which is in this case cholesterol, as well as presence of the single-stranded phosphatidyl tail. And the graph on the right shows you that when you just possibly add those compounds to neurons, you get reasonable uptake and silencing. So the hypothesis was that if this type of oligos can bind to the cellular membrane, they should be able to bind to their membrane of the exosomes. And indeed, if we take the exosome preparation and then we mix it with fully chemically stabilized sRNA which have no hydrophobic tag, you can see that all the sRNA stays in solution and this palette of the exosome is white. But in this case, sRNAs is labeled with a fluorescent label, so you can look at the pink color and see where the sRNAs is. But when we put a hydrophobic entity to the SI, we get very efficient association of those sRNAs with exosomes. Where up to, let's say, 60 to 80% of the compound added to the solution can be actually associated with exosomes. And then you can take those exosomes, apply them to the primary neurons, and you can see very nice delivery and silencing of the target gene. So the lower their points, the better degree of the silence. So if you look at those exosomes, what we see, we can see this under electron microscopy is those very typical bilayer structures. And when we try to map the location of the oligo, it's mostly associated with the exosomal membrane, but simultaneously you can detect some presence of sRNA inside the exosomes as well. So what was most interesting for us, which essentially was the scientific premise of this proposal, is that when we mix hydrophobic sRNAs with exosomes, we see around an order of magnitude shift in potency, indicating that when exosomes, when sRNAs are associated with exosomes, they traffic in neurons more efficiently than when they're not. So the next question which we wanted to study is try to characterize quantitatively how the amount of oligos added in exosomes affects exosome properties or not. And what was very interesting for us to see is that when we start to increase the ratio of sRNA to exosome in the loading mixture, we first reach saturation around, let's say, 2 to 3 sRNAs per exosomes. But then when you continue to increase the ratio, you start to see more and more sRNA kind of associated, become to be present in this nanoparticle form. What was important to see is that while increasing the loading ratio up to 10,000 did not change the number of particles, so they stay constant. When we increase the concentration of the sRNA per, or ratio of sRNA per vesicle, we actually start to see increase in the particle formation, which is indicative of the fact that in extremely high ratios, you start to see some artifact happening where the hydrophobic assays aggregates, and they potentially aggregate with some minor parts of the membrane's presence in the solution. So when you look at, when you take the 3 different fractions and you take the underloaded exosomes where it's below the saturation level, like optimally loaded exosomes and overloaded exosomes, and look at them on electron microscopy, they kind of look the same. But when we look at the efficacy, what we have seen is that both underloading and overloading result in significant decrease in efficiency of exosome ability to transfer compounds to the neurons. So for now, our recommendation is that around 2,000 to 3,000 sRNAs per vesicle is in the maximum. And while you quantitatively can load more, you start to interfere with exosomes' properties, and you also start to interfere with the vesicle ability to transfer inside the cells. So the way we can talk about the mechanism more, but I think the take-home lesson that too much of the good thing is not necessarily a good thing. So the number to remember is around 2,000 to 3,000 molecules per vesicle. So in a field of oligonucleotide therapeutics, a lot of great things have been happening recently. And in particular, the first 2 drugs got approved, and the most impressive things happening is expected approval of inclisiran with a fully chemically stabilized sRNA conjugated to GalNAc for treatment of high cholesterol levels, and those compounds are extremely important and durable in vivo in patients, and shows very limited adverse events. And the current NDA is filed with twice-a-year administration, so they have extreme duration of effect. And this duration of effect is really dependent on ability on full chemical stabilization of the sRNA. So what we decided to do, and full chemical stabilization is absolutely feasible. What you use by mixture of those metal fluoride modification, you can create molecules which are metabolically stable, but they efficiently load into RIS complex. Actually, in a standard in vitro assay, they load into RIS complex more efficiently than naked sRNAs, but most likely this is their artifact of enhanced stabilization. There is no impact on the RIS cleavage rate, and in tissue culture, those type of compounds shows equal potency to a naked sRNA. So you can metabolically stabilize a oligo in the way that it still very efficiently interacts with the RIS complex. But this type of stabilization has a profound impact on ability of oligonucleotides to be delivered in vivo and to be accumulated in the tissues. And what you can see here is just 2 different sRNA compounds. One is partially modified, like 65% of the ribose modified, and you can see that 24-48 hours post-injection, you practically have no substantial accumulation in the tissues, while when it's fully chemically stabilized, the amount of tissue accumulation of those oligos is dramatically increased. The way we look at accumulation of oligos in exosomes, as well as with different chemical modifications, fluorescence is great, but it potentially can be artificial, as well as presence of fluorescent labels have a potential to alter PK/PD behavior. So we always complement it with another assay where we directly measure the level of guide strand accumulation in a tissue. And the principle of assay is very straightforward. So what we make, we synthesize a Pna strand, peptide nucleic acid, which is fully complementary to the guide strand of Srna. And then because affinity of a Pna RNA interaction is extremely high, it competes out the sense strand. And then we can, and because the Pna strand is fluorescently labeled, we can on Hplc resolve and separate this hybrid Pna guide strand duplex and quantify amount of the presence of the guide strand in a tissue. And we call this assay Pna Hybridization Assay, and it originally was developed by Axel Labs and Roche, and there is a lot of publication in the literature which shows that it has a great dynamic range and sensitivity in a femtomolar range. So when we're using this assay, we quantified amount of the guide strand accumulation in the tissue when you compare a partially modified Srna and fully modified Srnas independently of type of conjugate you use and independently of route of administration, whether IV or sub-Q. Full chemical stabilization really confirms significantly enhanced tissue accumulation. So now with those chemically modified compounds, we can do a lot of fun things because we can change the nature of the conjugates. And this is one of the novel divalent structures where we can inject it not only subcutaneously, but we can inject it in CSF. And with single CSF injection, we get complete silencing in a brain at 1 month, 4 months, then 6 months post-injection. This work has been published, so if you're interested, you can look at it more. But the main point which I am trying to make is this full chemical stabilization of Srna with conjugate-mediated delivery seems to be absolutely essential for in vivo efficacy. So we decided to explore what is reasonable, what is the impact of chemical stabilization of Srna on exosome-mediated transfer. And the reason why we decided to do it systematically, because there is mixed reports. Some people have reported wonderful in vivo efficacy of exosomes-derived Srnas, but the claim is that it's only worked with non-modified Srnas, and introduction of Srna modification block exosome-mediated transfer. So what we did now is we synthesized, we started with a compound which has a phosphate, 5' phosphate. It's partially modified, and it works to some degree in vivo, both in Srnas and in exosomes. Then we modified all the ribose, and in vitro, the impact, you start to see some small increase in efficacy both with Srnas and exosomes. Then on top of it, we put 5' vinyl phosphonate modification. This is a modification which blocks dephosphorylation of the guide strand and ensures that the phosphate is always present, and phosphate on the 5' guide strand is one of the recognition elements essential for risk assembly. And while this modification in vitro have no impact on Srna only uptake, we have a measurable impact on exosome-mediated uptake. And finally, we mix all the things together where we apply 5' stabilization as well as remove all the riboses. And in this case, we does see approximately 5 to 6-fold enhancement in activity. So so our recommendation at this point, if you're loading sRNAs or antisense and exosomes, trying to ensure full chemical stabilization and trying to ensure that the 5' phosphate is stabilized as well, it's probably a good idea. So hydrophobic modification, and in this particular case, cholesterol, is something which is absolutely necessary for driving this membrane interaction. And there is a lot of publications from Roche and others saying that, at least in vivo, if you change the stability of the linker, you can change the efficiency of association and transfer. And what we decided to do, we decided to use a standard cholesterol linker, which is TAG. It's not fully stable, but it's probably very stable in the context of the experiment. Then we introduced 2-deoxy modification, which makes it labile but relatively stable. And then we introduced 2-RNA modification, which makes it very, very labile in a context of a great aggressive biological environment. So when we take those compounds, 3 compounds, which has a different linker connecting cholesterol, and looking at the passive uptake, as you can appreciate, there is no measurable changes. Passive uptake of sRNA alone, non-formulated sRNAs, they all look exactly the same. And the reason for that in those tissue culture experiments, we're using low serum media, and the uptake is extremely fast, like within a minute of exposure, you start to see a lot of oligo accumulated inside the cells, and within, let's say, an hour of exposure, you almost achieve a saturation level. So probably this changes in stability of the linker in those conditions has cholesterol is still staying attached, and as soon as the oligo is internalized by the cells, presence, attachment of this hydrophobic label is not essential. With exosomes, it's a completely different picture. It's like black and white. So when we made a completely non-stable linker, so there's 2 RNA molecules, 2 RNA bases, we lost activity. There's none. There's no efficient transfer into the neurons at all. When we make it a DNA, which is more labile, we reduce the efficiency, and our standard non-cleavable or not relatively stable chemical linker was the best. So in a context of loading sRNAs and exosomes, we will prefer to use a more stable linker. And I think the reason for that is when we think about exosomes, we kind of call it this fancy name, but in reality, it's biologically produced vesicle. So it has a lot of RNases and it has a lot of proteases. And whenever you're making this mixture, it's any non-stabilized bonds within the RNA will effectively get degraded, and as soon as you cleave, let's say, this RNA, hydrophobic label is gone, and then there's no efficient association with the vesicle and sRNA. So everything I have been talking so far about is related to the chemical nature of sRNA and the link to the hydrophobic group and impact on exosome loading. And everything has been done in a context of a cholesterol itself, which is very common part of like 20 to 30% of most cellular membranes are cholesterol made, but there's many other hydrophobic modifications. So what we decided to do, we decided to explore how the nature of the hydrophobic entity impact ability of sRNA to associate with a vesicle and ability of exosome to induce a sufficient transfer to the neurons. And to do that, we synthesize sRNA with a panel of different hydrophobic conjugates looking at different type of saturated and non-saturated fatty acids, looking at their cholesterol, these are oleic acids, different type of hydrophobic vitamins, and we also synthesize them in two different forms, by itself as well as a phosphatidylcholine amide, which is mimicking a natural form of those hydrophobic molecules circulating in a serum. So what you see at the bottom is Hplc traces, where all those compounds have been run in identical conditions, and the time of retention at a reverse phase Hplc column is indicative. It can be used like a proxy for the compound hydrophobicity. So as you can see, we cover the range of different hydrophobicity of the conjugates. So finally, we'll look at the impact of changing the hydrophobic entity to the on amount of sRNA loaded in exosomes. And what we can see is that cholesterol works well, but there are several other conjugates which actually gives us better association and better efficiency of the loading. When we put it on a curve and try to correlate the hydrophobicity of the conjugate to efficiency of association, it's almost 1-to-1, yeah. So there's a great correlation. So the more hydrophobic is the modification, the better is the association with exosomes and with DCA, PC tocopherol, and tocopherol itself showing better loading efficiencies than cholesterol alone. So finally, we kind of wanted to see whether there is any correlation between efficiency and loading in the exosomes and silencing efficiency, and each of those points is generated from a 7-point dose response, and you can appreciate that while DCA and PC tocopherol loaded very better than cholesterol, the efficacy was more or less the same, but loading with the tocopherol increased both efficiency of loading as well as efficiency of neuronal transfer. So far what I have talked about is trying to better define how to load sRNA cargo into the exosomes, and we discussed that you can use different hydrophobic conjugates, and from which tocopherol is the best, followed by cholesterol and DCA. It's preferred to have fully chemically modified as a full chemical modification and stabilization of sRNAs, which include both ribo modification as well as chemical stabilization of the 5' phosphate. And there is an optimal for the number of oligos associated with exosomes. Too much of the good thing is not great, because you start to get aggregation of the vesicles, which interfere with its biological properties. So around 2,000 to 3,000 sRNAs per vesicle, it's maximum effective loading efficiency. So the next question is how to produce exosomes. And I know that this is a topic of a lot of publications, and it's topic of a lot of discussions, and the paper actually was published in Molecular Therapy, so you you're more than welcome to read. But when we started in this area, we did exactly what everybody else was doing. We were using 3-layer flasks and growing a lot of cells to around 20,000 cells per centimeter in circumference, and then using differential ultracentrifugation to standard differential ultracentrifugation protocol to produce exosomes. And this process is very labor intensive and super inefficient. Just to give you an idea, to make enough exosomes to power a one well-designed in vivo study. So we're talking about maybe injecting around 40 mice. So we have 5 groups with N of 8 per group. We're taking 2 months with stocking incubators filled with those plates just to make enough exosomes to run this experiment. And we're not talking even about humans, we're just talking about doing it in mice. So clearly it was not the way to go. So, and this is where we enter in collaboration with Mass Biologics, with a non-for-profit branch of UMass Medical School, which is doing GMP manufacturing of vaccine, monoclonal antibodies, and DNA-associated viruses, and they told us that what we're doing is insane and we absolutely have to go to the 3D culture where we grow those mesenchymal stem cells attached to the beads and then essentially take the media and purify the exosomes by tangential flow filtration. And this is a protocol which is GMP compatible and it's currently being used for production of a lot of monoclonal antibodies, vaccines, as well as AAV. So when we took the same batch of cells and actually repeated this experiment 3 times, and we made exosomes from 2D culture and purified by ultracentrifugation and make it from 3D culture and purified from tangential flow filtration. Overall, I will say the peaks overlap. We probably get a little bit more resolution with tangential flow filtration, but the shocking result for us was that the number of particles or vesicles we got from the same number of cells in tangential, in 3-dimensional cell culture and tangential flow filtration was multiple orders of magnitude higher. So it's not 2-fold, not 10-fold, not 100-fold. It's like 10,000-fold more vesicle produced by the same number of cells. So when we look at ability of those TFF-produced vesicles to transfer oligonucleotides in neurons, it's not only we got more vesicles, they were at least an order of magnitude more potent in neuronal transfer. And finally, this enhancement in potency is related to the fact that we are loading more oligo in those vesicles. So it seems to be that the ultracentrifugation process is not only very inefficient in purifying exosomes, but it's also resulting in a structural and functional damage when it interferes with exosome ability to perform its biological function. And I will say that it might be the reason why there's so much discrepancy in literature between efficacy of exosome functioning in cis versus efficacy of exosome functioning in trans, because it's type of a purification which was at least originally widely used in many different labs, and it's considered to be kind of used to be a standard in the field. So from our experience right now, ultracentrifugation cannot be used for exosome purification if you're interested in function, because you're both getting super low yields, you're losing majority of material, but most importantly, this process itself, it's damaging to biological content of the vesicle and its ability to transfer to function inside the cell. Okay, so at this point, when we're producing exosomes, we're using a three-dimensional cell culture where we have optimized the protocols which allows us for mesenchymal and stem cells in particular, which allows us to use beads, and we actually, because we're an academic lab, so we try to save money. In GMP manufacturing, they never use the beads. We have reused them, and this is fine. You just sterilize it on the way and then recoat it. And then it's combined with tangential flow filtration, where we systematically evaluated the impact of the size of the filter cutoffs, etc., on yield purity ratio. And the paper is published in Molecular Therapy, so if you want to learn more, just go and read. We provided all the details. So finally is we wanted to see whether the composition of exosomes might be affecting trafficking. And the first experiment, which we did a long, long time ago, now it's like 4 years ago, we decided to see whether if we take exosomes from 3 different cell types, and in this case, we look at bone marrow-derived mesenchymal stem cells, U87 glioblastoma cells, and HUH7 hepatocellular carcinoma cells. And what we did 3 vesicle preparation on different days. And from each media we collected what we called exosome fraction, which let's say small microvesicles, and also microvesicle fraction, which is larger microvesicles. And for just convenience, I will continue to call them exosomes and microvesicles, but the only differentiation is in size. So microvesicles is a larger fraction, exosomes is a smaller fraction. And then what we did a quantitative proteomics and lipidomics. And with proteins, we were able to detect 35,000 proteins, and with lipids, we were able to detect around 2,000 species. And lipidomics was done in a collaboration with both pharmaceuticals. So this is the total data, which I do not expect you to kind of read and understand, but I wanted to make a couple of points. So first, we see very good consistency between 3 biological replicates. Yeah. So you can, this is unhierarchic, unsupervised clustering, and essentially vesicles derived from different cell types clustered together. The second thing, there is significant differences in both protein and lipid signatures of between exosomes and microvesicles. And again, exosomes is a smaller vesicles, microvesicles is a larger vesicles from a sake of today's presentation. So let's look in detail. So when we perform a pathway analysis, what we have seen is that there is multiple pathways which are co-enriched in each exosomes and microvesicles, which is vesicle membrane transferring GTPase activity, translation, and glycolysis. But there was also a pathways which were specifically enriched in exosomes only, like extracellular matrix, receptor binding, and immune response, and only pathways specifically enriched in microvesicles, which is like mitochondria, endoplasmic reticulus, and proteasome, which is probably is indicative that microvesicles are really enriched in fraction of degradome, or just vesicle fraction, which is responsible, like apoptotic bodies, etc. Another interesting thing is that there's some correlation between those enriched proteins from different type of exosomes, but there's also a lot of differences. So the next thing we did is we look at the degree of correlation between exosomes protein content and exosomes and microvesicle protein content and cells for different cell types. It's a complex slide, so I will spend like half a minute explaining it. But essentially what we have seen is that on a protein level, we have seen that exosomes derived from two different, that exosomes are dramatically more they're different from the cells. So while when you look at the protein composition of exosomes, it's very different from the cells, and there's more correlation between exosome to exosome rather cell to cell. While microvesicle seems to be more similar in content to the cells, so which is indicative that exosomes might have some active trafficking abilities or active differentiated functional content. And the second thing we have seen is that there are really strong correlation between exosomal content from two different cancer cells. So if you look at the proteins enriched in exosome from HU7 cells, which is hepatocellular carcinoma, and U87 cells, which is neuroblastoma, surprisingly, there was a lot of correlation. And when you look at mesenchymal stem cells exosomes and those cancer-derived exosomes, there's almost nothing in common. And on the right graph, what you see principal component analysis, which is just independently show exactly the same way that exosomes from cancer cells clustered together in mesenchymal stem cells it's a completely different beast. So what it tells us that the cell of origin might have a profound impact on exosome composition, where HU7 and U87 are similar to each other, while mesenchymal stem cells are completely different. So what was also very interesting for us is that when we perform the same analysis on lipid levels, the data is different. Now we see a lot of correlation in lipid content between mesenchymal stem cells derived exosomes and HU7, but no correlation between, much lower correlation between cancer-derived exosomes. And I think, and on top of it, there is no correlation between what rules derived from proteins and rules derived from lipid sorting, which indicates that the sorting mechanism responsible for enrichment of exosomes in proteins of lipids of different composition probably do not overlap. Okay, and finally, we kind of tried to derive kind of certain rules, and all the data is published. You're welcome to look at it, but it seems to be that EVs in general are enriched in a fatty acids with saturated with a longer and more saturated tails, which we have no explanation for, but it's just an observation. So what I said so far is that the answer to question is, does producer cells affect vesicle composition? Yes, absolutely and dramatically. And the second is that proteome and might be a way to distinguish, proteome of exosomes might be the way to distinguish between different producer cells and exosome origin. So the question is, so those changes in composition, does it affect trafficking? And this is where we heard from many people in the consortium that when you starve the mesenchymal stem cells, and majority of the work from now is focused on mesenchymal stem cells, that you can increase the amount of exosomes produced. We have not seen it, but independently what is the source of mesenchymal stem cells, when we starve them, we got exosomes which loads Srna more efficiently, and the exosomes which transfer Srna to neurons more efficiently. And this has been reproduced multiple times. It is very, very robust. Moreover, when we repeat exactly the same experiments for microvesicles, we don't see it. So it's really specific to exosome function. So this enhancement in stressed exosomes' ability to transfer material is coming from intracellular content, because if we treat it with proteinase K, we lose it completely. So it's indeed proteins on a surface of the exosomes which are responsible for it. And then we add the serum to those exosomes, nothing happens. Yeah, so it's really some changes in a protein composition. So then what we did was exactly what we have done before. We look at the proteomics and lipidomics, but now because we're focusing on a one cell type and comparing stressed versus non-stressed, we were hoping that we would be able to get more differences. So again, it's very high quality data, so 3 biological replicates, mostly reproduced. And when we look at the data, we have seen that there is a certain proteins which are absolutely enriched in a stressed those exosomes which are good in neuronal transfer. And when we look at those proteins, we came up with 7 candidates, and this is kind of the summary of the data. And the paper is now published in iScience, so you can look at it. So what then Rekha did, it's something only she can do. She actually manufactured all those proteins, and then we chemically meristylated it to ensure membrane incorporation, took the neutral liposomes and attached those proteins to the surface of neutral liposomes. And what we have seen that only 3 out of this list, incorporation in the neutral liposome, give us increase in ability to traffic compounds into the neurons. The next thing we did the same for lipidomics, and we have seen that there is one class of the lipids which was dramatically enriched in stressed exosomes compared to non-stressed exosomes, and they consist mostly of cardiolipins. With cardiolipins, you have the structure. There's 3 major components. It's a cardiolipin which has 4 tails, then it's monolysocarbolipin 3 tails and dilysocarbolipin. So what Rebecca did next, and as you can see, all of them, they're dramatically represented in stressed exosomes. And then what she did, she again took neutral liposomes and introduced 20 to 30% of the different cardiolipins. And she has seen that only introduction of dilysocarbolipin give you enhancement in neuronal transfer. And then what she did last is make this mimic of what she calls an artificial exosome. It's a neutral liposomes, which has 3 different proteins on a surface, as well as a 20 to 30% of dicardiolipin, and this artificial exosome is now perform very similar in ability to transfer compounds to the primary neurons as their original exosomes. What you see is this line in a black. It's just how the neutral lipid vesicles will perform, which we always include in controls because it's very important to understand that exosomes is lipid vesicles and there's some degree of the transfer. But I think it's very clear that the surface modification of those artificial exosomes is essential for transfer. And finally, we went ahead and did an in vivo study where we show that only when we inject liposomes formulated Srna, nothing happens. But when we inject exosome formulated or artificial exosome formulated oligos in vivo in a brain, we get a clear reduction in the Huntington mRNA expression. What I just wanted to finish with is the model where we believe that artificial exosome is a potential direction for engineering vesicles for efficient nucleic acid transfer, because of course it will require optimization of both lipid as well as the protein content. And also this lipid and protein content can be different for different applications. And with that, I would like to thank you for your attention and acknowledge a group of really talented and motivated chemists, biologists, and pharmacologists working together, and remind that a lot of work was led by Rebecca Kharasti, and this is a collaboration with Neil Aronin Lab in UMass, Marian DeFiglia Lab in MGH, and Berg LLC. And thank you for your attention.